Hey guys, in this video we're going to learn how proteins are modified post-translation. So we're going to see how and where proteins are modified and what type of modifications we might be making to our protein after we made it. So I'm sharing my screen with you here, and these are our slides from chapter 10 available on Blackboard. So let's look at how and where our protein might be modified. So remember, how did we make our protein? We used the process of transcription to make a messenger RNA copy. We used the process of translation to assemble the polypeptides, the amino acids, into a chain called the polypeptide. And then the polypeptides come together to make the protein. But remember, when your protein is forming, the polypeptide has to fold correctly, and then they have to stick together correctly to form your protein. So remember, the structure of your protein determines its function. So if it doesn't fold correctly, if it doesn't form correctly, then it's not going to work correctly. So it's really important to have the right amino acids in the right order, because if you don't, the protein will not fold correctly, it will not form correctly, it will be the wrong 3D shape, and if it's the wrong 3D shape, it's not going to work. So having the right amino acids in the right order is really, really vital for protein function. So remember that when the protein is folding, it's going to go through 3 or 4 levels of protein folding. First, we're going to start with our chain of amino acids. It's kind of a straight line of amino acids. It doesn't stay like this for very long. As it's coming off the ribosome, it actually starts folding right there, right then, into the secondary structures. So remember, when you're in the secondary level of protein folding, you're going to form coils, which are like this. They're going to be the alpha helix, or you're going to form pleats, which look kind of like this. They're going to be the beta sheets. So right away, as the protein is coming off the ribosome, it's actually starting to go into the secondary structure with the coils and the pleats right away. Now, when it goes into its tertiary structure, remember we said that's where you get this unique 3-dimensional shape. So we get lots of bonding between the OR groups, the radical groups of your amino acids, and you start to develop this very complicated 3-dimensional structure for your polypeptide. And then finally, when you get to the last phase, the quaternary structure, this is where several polypeptides come together to build your protein. So here you can see the protein hemoglobin, and you can see that it's made of 1, 2, 3, 4 polypeptides, and the 4 polypeptides are sticking together to form your hemoglobin protein. So a protein is usually 2 or more polypeptides that stick together. Now, a lot of the time when you make your proteins, they're very often synthesized in an inactive form. So say, for example, if you were in the digestive system and you have an enzyme that's a protein, and its job is to digest carbohydrates, right? So you have this enzyme, its job is to digest carbohydrates, but remember, you have carbohydrates on the surface of all your cells. So do you want to digest your cells, or do you want to digest the food that's in your digestive system, right? So very often enzymes are produced in an inactive form, and they're only activated when they get to their site of action. So this makes a lot of sense. You don't want to have an enzyme that could damage your cells activated too early. So usually in the digestive system, we make the enzymes in places like the pancreas, and then they get shipped down to the digestive system, and then they only get activated when they reach the digestive system, when they're in the area with the food, because we don't want to damage the cells of the pancreas by having an active enzyme in there. So very often your proteins are synthesized in an inactive form, and then they're going to be processed to become active later. So remember, your rough endoplasmic reticulum, it has got ribosomes all over the outside, and the rough endoplasmic reticulum is where most proteins are going to be modified. So let's look at the 3 ways that they could be modified. They could be cleaved, which means chopped up. They could be phosphorylated, which means we're adding phosphate groups. Or they could be glycosylated, which means you're adding sugar. So remember, way back when we were learning about plasma membrane, we learned about glycoproteins, glycolipids, right? So a glycoprotein is a protein that has a sugar attached to it. So adding that sugar, that would happen in the rough endoplasmic reticulum. So if your protein gets phosphorylated, it usually, phosphorylation is another way you can activate your proteins. Usually if your protein's unphosphorylated, it's inactive, and then when you phosphorylate it, it gets active. And then sometimes proteins are made and they're too large, so they're not functional because they have extra pieces stuck onto them. But when you chop off the extra pieces, they become functional. So like I said, a lot of enzymes that work in your digestive system work this way. So let's take a look at the hormone insulin. Insulin's job is to keep your blood sugar at the normal level. So if you eat a candy bar, your blood sugar spikes, right? So here's your normal blood sugar. It's about 70 to 100 milligrams of sugar in your blood. And then when you eat the candy bar, which has lots of sugar in it, your blood sugar level goes up, right? So high blood sugar is dangerous. This is why diabetics have to get treatment. You can't walk around with really high blood sugar. It damages your cells. So in a normal, healthy person, we have this hormone called insulin. The insulin takes the sugar from your bloodstream and it puts it into your cells. So when you have insulin, you can move the sugar from your blood into your cells, from your blood into your cells. And if you take all the sugar and put it inside your cells, then your blood sugar will come back down to the normal healthy level. So this hormone insulin is vital for normal regulation of blood sugar. So if you're diabetic, you either don't have insulin, your pancreas can't make insulin, or sometimes you can't, even though you have the insulin, you can't open the channel here that allows the glucose to go into the cell. So there are different types of diabetics out there. But the key thing is that when we make insulin, when your pancreas makes insulin, it is synthesized in an inactive form. So here you can see what the insulin looks like when it's synthesized by ribosomes inside the pancreas, inside the special cells in the pancreas. It's one long chain, one long polypeptide chain. Now, you'll see that when it's time to activate the insulin, you're going to chop it up. So remember we said cleavage is the proper term for chopping or cutting up your insulin. And you'll notice that you cut here and you cut here. So you're cutting off that entire section of the polypeptide chain, and then you've got the activated insulin, which is much shorter. It's actually two short chains that are stuck together. So your insulin is floating around in your bloodstream and it's inactive, and then it becomes activated at the appropriate time by getting cleaved or by getting chopped like this. So this would be an example of a protein being modified after translation. Chopping a bit off, adding a sugar, phosphorylating. These are all ways that you could activate your protein after you made it. So that's the end of our video about post-translational modifications of proteins.